All right. Hello, everybody. This is a Galaxy BRC Analytics.org webinar. We've been doing one or two already. This project has started in September of 24. So we've been, just a few months in, well, 5 months in. So today I'm going to be talking about what you can do already and what you will be able to do in the next few months. And we are kind of in the part of this project where our functionality increases, I would say, and hopefully in geometrical progression, is that more and more things will be able, people will be able to do more and more things in the coming months. So today I divided this into, well, in two parts, well, really in three parts. I'm going to remind everybody what BRC Analytics is, why it exists, what it's going to do, and in general where we're going. And then I'm going to highlight two kinds of analysis that you can do with workflows within this framework. One will be dealing with analysis of transcriptome contrasting drug-resistant and drug-susceptible strains in Candida auris. And another one will be dealing with large-scale surveillance of variants in Plasmodium vivax using a public data set. So let's start with BRC Analytics. What is it? So the key challenge, or at least it seems to us, the key challenge, it seemed like a key challenge to us when we wrote the proposal. And again, please use the chat functionality to not only to ask questions, but also if you think that we're getting things wrong and maybe we should consider our direction, please. So use this as a suggestion as well, because believe it or not, we actually read those things. So I think the key challenge for all life sciences, not necessarily for anything that involves infectious diseases or pathogens or hosts or vectors, is interpreting results in the context of existing data, because it can be very confusing to know what exists, where it is. And most of the analytical challenges are related to, well, let's call them stupid problems. How do I map, gene names from this build to that build? How do I change formats and things like that? So it's really not a joke to say that the key challenge is, in fact, yes, interpreting results in the context of existing data, knowing where this data is, and being able to take full advantage of it. So generally, most of the data we analyze on a daily basis when we're trying to interpret our own results, we get from public repositories. And these repositories, there is a big umbrella organization called Global Biodata Coalition. There is also the three resources that most of you know about, NCBI, the National Center for Biotechnology Information. It's a part of NIH, part of the National Library of Medicine specifically. And this organization works in concert with the European counterpart, EBI, and with Japanese counterpart, DDBJ, DNA Data Bank of Japan. So these resources maintain, of course, literature. They maintain all sorts of data, ranging from genomic sequences to expression counts for different genes in different organisms. So these are official sources of data, and within the scope of BRC Analytics, we're trying to base everything off of these official sources of data. We are trying to make them more usable for end users. We are trying to make it easy to access data from there, and we'll make it easy for you to interpret your results in the context of the data which is already available. Because remember, any organism you're analyzing, you probably need to have a reference genome, a set of gene annotations, things like that. And so for vast majority of organisms, this data is already there. You just need to know how to get it. On the other hand, there are interpretation frameworks that connect to these sources. such as, for example, UCSC Genome Browser. It's a resource at the University of California, Santa Cruz, which pulls the data from these resources and provides you with a canvas on which you can visualize and interpret your data. There are also frameworks like Galaxy that allow you to run thousands of tools and hundreds of workflows on a free infrastructure provided for you to, again, interpret your data in the context of the data sets from these places, or to reanalyze existing data, things like that. So this is the general idea. So the key question again is there a way, is there a convenient and intuitive way to obtain public data about my species of interest? Say I'm studying avian influenza or Plasmodium or a host or a vector of any kind and so on. So what we don't want to do is we don't want to duplicate existing resources. We instead want to come up with new ways of gluing them together and really enabling you to either reanalyze existing data or analyze your own data in the context of what's already there. So what is BRC Analytics? It's a consortium of these kind of four main stakeholders. There's a Galaxy project. This provides analytical capability. You will see today that this is a platform which allows you to run tools, combine them into workflows, so do essentially any kind of analysis you can imagine with any open source tools that exist for genomic and general data science. We're not limited to genomics, really. In doing this, we rely on data from NCBI. In particular, we have very good, tight interaction with NCBI datasets team, with NCBI Comparative Resource Group. We also have existing collaborations with NCBI SRA and other places. NCBI, of course, not technically part of this in the sense that, so this is it's a funded project and it's funded by NIH. So I guess NIH cannot be a part of NIH funded system, but we're collaborating with NCBI very closely. Another part of this consortium is UCSC Genome Browser. It's an integral element of this system, and we use it to visualize existing data, to give you an opportunity to visualize your results in the context of existing data. And it's also an ideal platform for adding custom annotation. For example, a lot of communities, for example, communities that in the past relied on ViewPathDB, they created a lot of custom tracks or custom annotations because they possess unique knowledge, for example, about Plasmodium genes or genes in other organisms. So UCSC is a good way that would allow adding this custom information on top of what's provided by reference sources. Again, reference sources such as NCBI. The key difference between this resource and other resources is that we allow you to do analysis actually, meaning that we allow you to run complex computational pipelines. So today I'm going to show you, for example, reanalysis of 1,400 samples of Plasmodium vivax. In order to do that, you need a place to compute, because you need a place where you would upload your data, where you would run your tools, where you would interpret your results, and so on. And so that infrastructure is cloud-based, but it's a public cloud. It's a cloud which is provided by Texas Advanced Computing Center, and it's a part of a larger access consortium, which is essentially a constellation of large supercomputing centers in the US. So it's a cloud, but it's a free public cloud, which is designed specifically for scientific inquiry. And it's a very large and robust resource on which you can do complex analysis. So, as I mentioned in the beginning, we're just starting. So it's been a few months, and I know that some of you who are on the call, you are continuously going to our site and looking and seeing what's new. So We know that we don't have all features yet, but we're slowly going towards that goal, and so we're getting closer. So let me just give you a brief overview of what we have at this very moment. At the end of this presentation, I will show you what we'll have in a few months. But I have this problem that sometimes I forget to switch tabs in my Zoom. So if I'm talking about something and it's not what I'm showing, please let me know, so I don't have to do this again. But I think I switched this now. So this is the BRC Analytics as it looks today. Initially, we ingested all the organisms that were present in ViewPathDB, because ViewPathDB had a list of organisms, so we ingested all the same set of species. So at this point, we have two concepts in BRC Analytics. We have organisms, and let me remove these filters. So this is the current list of species that we have. And usually, if for those of you who are familiar with how everything sort of revolves around genomics, but in terms of so you have an organism, and for a given organism, you usually have multiple versions of the genome. And these multiple versions of the genome may be because organism has, for example, multiple strains. So for a given species with a specific tax ID, you can have multiple associated genomes. It's also possible, for example, in cases like human, is that you just this is how it is how it evolved. You have multiple versions of the same genome, which reflect different stages of evolution of sequencing technologies and so on. Of course, you may ask, why don't we just keep the latest version? Well, latest versions sometimes are more complete than the previous versions, but they usually contain less annotation, because ultimately when genome versions change, you can view genome as a coordinate system. So once they change, your coordinates change. And so it's not that straightforward to actually lift a set of coordinates corresponding to, say, genes from one assembly to another assembly. So this is why usually we have multiple assemblies per species. And again, this is the list as of today. However, we at this point we were conservative into not including many more species, but ultimately we probably going to have everything that is in the CBI listed here. So we actually want to ultimately expand it to all public genome data, whether it's pathogen data, host data, vector data, anything. So let's use an example. So today I will use Candida auris as one of the examples. It's one of the pathogens of interest due to its emerging antimicrobial resistance. So suppose you're interested in this particular bug. So you would search here. You would find that we do in fact have that species. And so this, in this particular view, maybe I should make this larger. You see this is the organism tab that is active here. But if you select on the species, you will see that which assemblies are available for this organism. So in this case, for ours, you have 6 assemblies. Usually, the sources of truth, such as NCBI, designate one assembly as the reference. And so here there is this column called, is this a reference? No, yes. So there is one reference, which is sort of an official reference genome for this particular species. So I'll expand on this in a second, but there are some other columns in this view, and the way we present this information will likely change. But the bottom line here is that you can see what level of assembly it is. So of course, the highest level is a complete genome. Then you can have scaffolds. Some assemblies are probably in contigs. But in general, when you have genome such as a complete or at chromosome level, that means it's a higher quality assembly. And there are some metrics such as total genome length, number of scaffolds. So the smaller the number of scaffolds, the better the genome quality. Ultimately, you want to have your number of scaffolds to be equal to the number of chromosomes. And it also lists additional statistics about these genomes, such as N50, which is a measure of completeness. In this case, the higher the number, the better your assembly. Coverage, and for example, does this genome have annotations or not? So again, which one you're going to choose here depends on what you're doing. If you are doing some new analysis and you want to, for example, understand, I don't know, you're doing a genetic variation surveillance, I would probably choose a reference genome for this case. But in the example that I'm going to show you, the data was generated specifically for this strain. So we're going to be using that assembly in this particular case. But again, what I want to emphasize right now is that essentially what this site is at this point, it's an organism and assembly browser. For each assembly, you have various resources, various external resources where you can go and get more information. Such as, for example, NCBI datasets, NCBI taxonomy. And for each of these organisms, we connect to a genome browser. We use UCSC genome browser for this purpose. So in this case, I'm, for example, looking at that particular B8441 strain assembly of ours. You see that it's not a particularly dense browser in terms of how much information. You really only have 2 gene tracks here, the predictions plus the annotations lifted by homology, so from RefSeq. And again, this is an area where we need your help because, for example, if there's anybody on this call who works with this particular yeast species, if you know additional resources that need to be put in here, please let us know, because it's very easy for us to ingest additional data. So this is, again, why we have sort of this dichotomy. On one hand, we rely on NCBI because this is the source of reference data. But on the other hand, this is why we partner with UCSC Genome Browser, because it's easy to add custom tracks which are, for example, represent domain knowledge from a particular community, from our community or for any other organisms. We are in the process of moving some annotations that were present in ViewPathDB. So in case of Plasmodium, various Plasmodium species, you will see, we're starting with falciparum in this case, you will see an increase in the number of tracks that we will provide. But it's one of the questions I have for you is if you know of additional tracks for organisms that are included in BRC and these tracks are not there, let us know. We will ingest them. We did not get a lot of requests for this yet, but hopefully this will be changing, and I'm really hoping that we'll get to the point that we will need to establish a certain procedure, a form that you will be filling. But at this point, just reach out and we'll add them. Okay, so the general idea is that we have organisms, we have assemblies, and in this list of external resources, we're going to have more. So it's just an initial set at this point. But for example, again, if you know of established external resources specific to a particular organism, we can add them here easily as well. So perhaps one of the main messages of this webinar is that we need your help too. because we obviously are not specialists in all of these organisms. So we really want to get community buy-in here. Again, we're new, so it's easy to change the direction, change design. So use this opportunity, because as the project progresses, of course, it will become more and more well-defined. But at this point, we still have a lot of room for perhaps alternative views, things like that. Anyway, so we have, as I mentioned, a way to look at external resources. We also have this Analyze button. So if you click on this Analyze button, you will have a list of actual data analysis workflows that you can run on this genome, in the context of this genome. Because every time you're doing, I guess, standard types of experiments, transcriptomic experiments, so different types of RNA-seq, bulk RNA-seq, single-cell RNA-seq, types of regulation surveys, ChIP-seq, cut and run, permanganate-seq, any kinds of resequencing studies like variant calling, for example. You need a reference. So you do this in the context of a genomic sequence. And so here, when you choose an organism, choose an assembly, and then click on Analyze button, you essentially get a list of things that you can do in the context of this genome. You will see that it's not a very big list at this point. I will, at the end of this presentation, I will show you our kind of a roadmap on the types of analysis that you will be able to do here. But you can see that at this point, it depends on different organisms have different types of analysis, because if you have a virus or if you have bacteria, there's some analysis which makes sense in this context, but that don't make sense, for example, for eukaryotes. So there's a difference between what you can do on any particular assembly. But in this case, for example, you can do variant calling. Just one possibility here. In terms of transcriptomics, there are more possibilities. You can do different kinds of RNA-seq, the bulk RNA-seq in this case, and single-cell RNA-seq. You can do different kind of regulation analysis working with proteins that bind to DNA in this case. So that's just what's possible right now. And maybe you're listening to this and so maybe you're starting to get a little bit confused. So let me show you how this works. So let me go back to my slides. And we were, well, before I do that, let me mention one more thing. So what will happen in the next few months is that in addition to these tabs that I'm mousing over here about organism assemblies and roadmap, there will be an outbreak tab, and that tab will point to organisms. It will be essentially a filtered down list of organisms that are viewed by current the public health authorities as organisms of priority. So what essentially NIH thinks is the current list. So it will be an, again, outbreak tab, and there will be workflows specific for these organisms. Okay, let's go back to slides. So imagine the following scenario. So
Anton, can I interrupt real quick? Somebody asked a very reasonable question looking at the list of workflows, saying, what's the use case for an assembly workflow, given that your entry point is an assembly?
Excellent question. So right now, all the entry points on BRC are via a particular genome, but that's not going forward. There will be an entry point where you just want to do analysis and you want to do assembly, and you want to run analysis which are not hooked on a particular reference, such as assembly, for example. And another area which I ignored completely at this point, there is also BRC will also allow comparative analyses. So this is when you want to compare two existing assemblies, or when you want to compare an existing assembly to something you would assemble yourself. This would progress in two stages. At the initial stage, because it's a hard problem, at the initial stage, we would allow pairwise comparisons. So for example, you want to compute, you want to establish synteny between two genomes. And once this is done, we will expand to multiple genomes. So essentially, what will be happening behind the scenes is that we will allow people to run genome-wide alignments. There are some interesting performance and usability questions here. Is that for smaller genomes, like yeast, for example, or Plasmodium, or some insects, where there's plenty of not so large genomes, you can probably run alignments yourself. It's still going to take some time, but it's a finite time. In some cases, when you want to create large, when you're working, for example, with host genomes with mammals, and you want to generate multi-way mammalian alignments, this is a little bit a different problem. Places like UCSC already generate alignments for vertebrates, and we will leverage that. But we also need to account for cases when people want to create multiple alignments from scratch based on something that they want to study. So this is coming, but I'm not talking about this yet. I hope this will be the subject for the next webinar. But that's a great question. It means that you're actually listening what I'm saying. This is great. So here's an example. I was looking at the list of current pathogens of high importance. So Candida auris is one of them. And I was interested in what people do, what kind of analysis people do, what kind of transcriptomic analysis are done. So one of the analysis that is described in this paper, this is not a very new paper. The paper is about 2 years old. But essentially what's happening in this case is that they are contrasting expression profiles for susceptible and resistant strains of Candida auris. And so when you look at the paper like this, you might want to, replicate what they've done. Maybe you want to reanalyze what they've done, or maybe you want to see what they've done and use and apply this to your data. So in what I'm going to show you today, I will reanalyze this, essentially reanalyze their study using the BRC Analytics framework. So as most papers of this type, there is actually not that many RNA-seq studies for hours, so this is one of the more comprehensive ones. But usually when you do surveys like this, you read the paper and you get to the materials and methods sections, and you get something like this. This is data available from here. So you click on that, and you're probably going to get to some kind of NCBI page like this. And this is GEO, this is Gene Expression Omnibus. I should mention that one of our requests from our funder, from NIAID, and in general, one of our goals is, if you remember when I was showing the list of external resources, so these external resources were taxonomy, UCSC genome browser, NCBI datasets. We're also gonna hook the geo data sets for a particular species there, so you can pool the data. You can see all the data sets that are available from geo for a given species, for example. Okay, so you get this, and what I want to do in this case, I want to actually rerun RNA-seq. So usually you look around this page and you will find actual data the actual, in this case, Illumina actual underlying data sets for this particular study. As most studies, there is a bit of a confusion here because the paper says that it was paired end data, but they submitted it as a single end data. So there's always like this, and not all the samples are labeled correctly, but I'll leave this on the conscience of the authors. So you actually can see I'm not going to move this data. Of course, I'm just going to show you how this works. It's a sizable data set, so it's 15 samples. It's a nice data set because for each isolate, it contains replicates. So you actually have 3 replicates per data point. And you can see that if you select these data sets, you can move them to Galaxy. So BRC Analytics is essentially a data browser on top of the Galaxy system, which allows you to run these analyses. I already pre-uploaded this data into Galaxy. I cannot show you everything within 1 hour, but at the end of the presentation, I'm going to mention that we are starting a series of screencasts of short videos, which will detail how we do these things. This is coming in April. And considering that, I judge by myself, I don't have a very good attention span. So these videos will be short. It's not going to be an hour video how to do RNA-seq. Instead of it will be probably 20 videos on how to do RNA-seq. Each video, up to 5 minutes long, how you, for example, upload the data and do things like that. So normally, here's how you would be able to do this kind of analysis via BRC Analytics. So you would start at the BRC Analytics site. You would select, well, I already have hours. Let's do this again. I would have this. Because, again, I read the paper and the analysis that they've done against B8441, and it's a decent genome. It's not referenced, but it's actually in good shape, and actually some of its stats are better than for the reference. So this time I'm going to click the Analyze button, and I'm going to select variant calling workflow, and I'm going to click this Launch in Galaxy button, which would bring me to this tab. Let me make sure that. And so in this tab, you can see that because I started from this page, so I started with a page which shows me the particular genome that I'm working with. So when I actually go back to Galaxy, you will see that my gene annotation is already pre-filled to the right version. My genome is already also pre-filled to the right version. So all I need to do is to move the, let's see, is that the right data here? Yeah. So I need to move the right data into my, into my, what did I do wrong? Okay, so I'm sorry, stupid me. So I selected a wrong workflow. So for everybody who's watching, live demos are dangerous. So it's not BRC Analytics, it's not Galaxy, it's me. So let's do this again. I'm gonna start in BRC Analytics, click on organisms, select hours, And of course, what I want to do on this assembly, but I don't want to do variant calling. I want to do transcriptomics. This is a transcriptomics study. So stupid me. I have 4 workflows from transcriptomics. This particular study that I'm talking about is so it's a bulk RNA-seq. So let's proceed with the bulk RNA-seq idea. And in this, it's a single-end data because despite the fact that the authors say it's a paired-end data, they actually submitted it as single-end data. And that happens all the time. And the only thing I want to say here is that when you submit your results, please make sure that your students fill that form correctly. Anyway, so I'm going to click Launch in Galaxy. That will throw me in the Galaxy, same interface you've seen before, but now it actually recognizes the data because it's a single-end collection in Galaxy. So it has 15 data sets, just like we've seen in Geo. And so what you see here is a workflow run interface in which you really need to, the only thing you need to do is to supply the data. It could be data which is pulled from SRAs in this case, or it could be your data. I'll ask it to generate additional QC reports. The information about genes is already pre-filled. Information about the genome is already pre-filled. So all I really need to do at this point is to click Run. And so this is when you're going to see several things here. You're going to see the workflow that you're about to run. It's a workflow which takes your reads, maps them against the genome using, in this case, star mapper. And ultimately, it goes and generates, so once this is all over, it's going to generate count tables. So read counts per gene for each of the samples. This entire thing runs at the TAC, the Texas Advanced Computing Center that I introduced in the beginning. And this is an example of a relatively small workflow. It's only a relatively small analysis. I mean, it's only 15 data sets. The analysis that I'm going to show you in a few minutes analyzes over 1,000 data sets. But the basic idea is that you go to BRC, you select the genome you want to analyze, you pick the workflow you want to run, you start it, you supply it with the data, and it starts running. One of the things that will be happening shortly, it's already in our test site. So there's an instance of BRC which we use for testing. It's going to have a different view of. It will allow you much more flexibility in uploading your own data. So ultimately, after all of this is done, and despite the fact that it's not a very big analysis, but will probably take, I don't know, 15 minutes for this to run, I, of course, pre-computed these results. So let me show you how it ends up. So at the end of all of this, you will be able to see the list of downregulated and upregulated gene. And in this particular analysis, I've done just one comparison. So if we go and compare it with the results from the paper, so I've only done this analysis, but you can see that it's essentially the same set of 3 upregulated and however it's 10 or 12 downregulated genes. One of the challenges in these analysis, of course, is the gene name mapping. And again, this is one of the areas where we would need community help for identifying gene names mappings. But you can do this. It's you can take a data set of any size, and you can do, for example, transcriptomic analysis, as I'm showing here. So that's one example that I wanted to show. Another example deals with analysis of Plasmodium vivax data. So there is this effort called Malaria Gen, and it deals at this point with two Plasmodium species, with Falciparum and with Vivax. And
Anton, before you move on, Peter asks if in the future, if we want to share results with each other via BRC Analytics, is the solution to publish to Geo? Because he's thinking about, building up a knowledge base over time from all these different results. What is our thought on how to share results, kind of with the wider world?
So Geo, I know that there is a, so is Peter asking about the results of a particular investigator, or in general about all Geo data sets? Because NCBI tries to convert some of the Geo data sets into actual gene counts, but they're doing this only for human and mouse. So there is nothing that kind of prevents us from, for example, if there is a community request, is that for, say, this species, there is a fundamental paper that does expression analysis. It's the most important paper for this organism. Can you please convert this into gene counts and distribute it via BRC Analytics? Of course, the way this will work is essentially we are going to provide this information as a track in the browser, because we control that browser, so we can add any tracks to it. And when you invoke a workflow from BRC Analytics, when you click this Launch in Galaxy button, you will be able to choose this track as one of the data sets you might want to interrogate. So you will be able to look at this data using the browser, and you will also be able to use this data in your analysis. For example, if it's a gene track, you want to run RNA-seq analysis and you want to compute your counts not against the, say, NCBI gene track, but against some custom gene track. Does that
so I asked Peter to unmute if he wanted to elaborate or comment on that.
Yeah, Anton, I don't know if there's a way to squeeze it into the browser essentially for you, but imagine that we've got two different, I don't know, imagine we've got a matrix of things. We've got different drugs, we've got different life stages of the parasite, and I want to see what is known about expression experiments in terms of these different experiments. And it would be nice to not redo from scratch every time.
So going forward, I think I have a slide on this somewhere. So this is not present on the production site yet, but you will be able to pull into analysis any tracks which are associated with this species. So any tracks that you will see on the browser, you will be able to, when you choose a workflow and you're configuring your run, you will be able to say, okay, I want these genes, or I want, this set of peaks, or I want this set of gene counts, things like that. But you need to tell us what you would want to include here. So that's the mechanism. So another analysis I wanted to show, sharing this tab, we have 10 minutes left, is the reanalysis of Vivax data. So for this, this data set here, I thought there's a link here, but so it's a public data set of 1,800 samples. Well, in reality, it's less than 1,800 because some of them don't have very high quality. So when you actually QC this data, you end up with about 1,400 samples. So I wanted to use this data set to demonstrate the fact that what you can do in BRC Analytics is scalable in terms of that you can actually run large analyses. So another thing is that, which actually surprised me a little bit, is that Malaria Gen uses pipeline which is based on GATK, which is not exactly an ideal tool for calling variants in this particular pathogen. So I just wanted to reanalyze, essentially rerun their data using a more appropriate variant caller. And the way I've done this was like this. So I went to BRC Analytics. I chose, well, let me do one thing here. Okay, so I went to BRC Analytics. I clicked on the organism. Okay. And so in this case, that's the reference. In this case, I chose the best assembly here, which is designated as a reference by NCBI. Clicked on Analyze, selected a variant calling workflow. That's the mistake I did before. So this time I actually want the variant calling. Clicked Launch in Galaxy, and that gave me so this will eventually load the workflow run interface. And you can see that I preloaded the Plasmodium vivax data. So it's a full set of 1,400 samples. And the way I did this is I simply downloaded the list or the data list from Malaria Gen. And then Galaxy has a way of pulling this data in and presenting it as a collection that you see here. So all I need to do is to get the data, invoke the workflow, tell it what the data is, it's already here, and run it. So this will generate a substantial number of jobs because it's a multi-step workflow. So at the end, it will probably be about 12 or 15,000 individual analysis. But at the end of the day, we're gonna get a history. It's gonna take a little while to load it because it has close to 30,000 data sets. But we are in the process of taking this data, reanalyzing it, and comparing it with what Malaria Gen generated. I can just show you sort of a quick preview of this data. So in Galaxy, you can run a complex workflow like the one that takes, for example, 1,400 data sets. At the end, it generates list of variants. These lists of variants can be combined. So you get essentially a tab delimited report file. And then you can do various things. One of the things that you can do is you can write a custom visualizations. That's what I did here, because Galaxy includes things like Jupyter and RStudio. So, and I'm showing the wrong tab here. So this is my Jupyter instance running on Galaxy, which visualizes non-synonymous variants in this data set by chromosomes. So I have chromosomes as individual plots here. I have 7 of them. And I have the variants, the number of, so the y-axis in this case is allele frequency. So I have, interestingly enough, some number of variants with intermediate frequencies, which are not fixed. They're colored by how many samples they're present in. So again, I have 1,400 samples. And so most of the variants are not present in all samples, but I have hot spots like this. This plot shows only non-synonymous variants. And most of them are homogeneous in terms of it's one change, which changes one codon in a one way. But there are situations where there are multiple hits at the particular codon. So let me switch back to Galaxy, and this is the result of this analysis. So you can see that it's Galaxy workflows in this case was 40,000 data sets, because it's 1,400 samples, multiple steps of the analysis, and in the end, I essentially have my final result, which is a list of variants. It's a large list. It's 150 million variants before filtering. And you can see that they're classified as synonymous, non-synonymous, and so all the various ways in which I can interpret these data. I want to leave at least 1 minute for questions, so let me quickly jump to what's coming. So what is coming? So I understand this magic. The problem with doing short webinars is that I'm jumping across tabs. And if you really want to understand how this works, I'm probably not spending enough time on each and every step. And as I mentioned, we are going to start creating a short videos on these. And this will start coming out in April. We will start with these two specific analyses, with transcriptomics and with surveillance style variant calling. So it will really be digested to the point, okay, I read the paper, I get the list of accessions, how do I get them in the Galaxy? How do I prepare them? How do I figure out which data sets are good and which are not good? How do I start my workflow? How do I interpret whether my workflow is successful or not? How do I take the final result? All these steps will be individual short videos on each. So we will have more workflows. In Galaxy, there is a kind of an organization called Intergalactic Workflow Commission, and it's a place where Galaxy workflows are being submitted. You can get to this website today at iwc.galaxyproject.org. It's a new effort, but it already has a substantial number of workflows for various types of applications. We will have a better way to look at the data, and I can quickly show you how this will look like. So we will have kind of a graphical, again, what I'm showing you right now is a dev site. It's not for public use yet. It's our sandbox, our playground. But in addition, you will be able to essentially select your organism using either a list or this interactive viewer where you select a particular clade, and then it will get you to the list of genomes associated with that clade. So at least like this. You will be able to, in the workflows that I ran today, my gene annotations, so in this case, I did RNA-seq, so I needed to generate counts for each gene. So I need to get that gene information from somewhere. Right now, it's fixed to just the NCBI genes, but that's not the way to do it, because users should have a freedom of selecting whatever gene track they think is necessary. So you will be able to control which tracks you feed into your workflows. We will also, in the examples that I showed you, I pre-downloaded data from SRA. I did this via Galaxy. This is not ideal. You need to be able to query SRA directly from BRC Analytics. This is also coming. So you will be able to enter organism or project ID or geo-identifier, anything like this, to get a list of existing sequencing data sets and pull them into your workspace. This is probably going to be the first ever direct interface for SRA, which couples SRA querying with analytics. And I think I have 3 minutes left, so maybe it's a good time for any questions.
Okay, I'll start with this one. Indrish asked if there are Python Jupyter notebooks predefined for an analysis, so that users can just run it and customize it easily, or is it like just a generic Python notebook that then the user would have to figure out, what to do with it?
So the Jupyter notebook can be a part of the workflow. So the workflows that I ran, it's just a chain of normal tools. So for example, at the end of the variant calling workflow, what you get is essentially tab delimited data sets of variants. But we can modify this workflow so the endpoint is actually already pre-filled Jupyter notebook, which can be used to generate a particular visualization that I was showing you. So I guess yes and no. Yes, it's possible, but we don't have Jupyter notebooks for these workflows yet. Right now, when you instantiate Jupyter in Galaxy, you just get a default Jupyter image, which again does not prevent us from sharing notebooks specific to particular visualizations.
Okay. So yeah, there are no other questions in chat. So we'll ask if anybody else has any questions. You can raise your hand, or you could ask in chat.
Do you still hear me? Because I'm having some browser issue here.
Yeah, I can hear you just fine.
Do you see me?
No.
Okay, well, anyway, I have a hand and arms, things like that. Yeah. Okay, well, chat has many questions, actually. Oh, well, nice that you chose Vivex. Well, thank you.
Yeah, I think I'm reasonably sure we've just addressed all these questions that were in the chat.
I also need to thank Marius for alerting me why things weren't working. Of course, I chose the wrong workflow. So, and I really appreciate the support from BRC team. So as you know, the BRC is a consortium of Pathogen Data Network, PDN, BRC, and us, BRC Analytics. And I'm glad that they joined. Thank you.
Okay.
All right. So
I think we can wrap up.
This is recorded, so we'll share the video, but shorter videos are coming. Thank you very much.
Thanks, everybody. Bye.